Hello and welcome to the fourth lecture in our series on cell division. Previously, we covered the basics of Preprophase. In that, we had a detailed discussion on Phragmosome, what is a Phragmosome and why it is important. We had a detailed discussion on Preprophase band, what it is, why it is important. And in this video, we will be discussing Prophase. The cell gets ready to divide during the prophase stage, which comes after the G2 interphase stage. This phase is characterized by the cell compacting its chromosome into more compact state and the beginning of the process of mitotic spindle formation. Throughout interphase, the genetic material in the nucleus is composed of loosely packed chromatin. At the start of prophase, chromatin fibers come together to form separate chromosomes that are usually visible through a light microscope with a high magnification power. At this stage, the chromosomes appears to be long and thin threads. There are two chromatids on each chromosome. The centromere is where the two chromatids meet. During prophase, gene transcription stops. It starts again in late anaphase or early G1 phase. During early prophase, the nucleolus also disappears. Centrosomes are structures in animal cells that are close to the nucleus. They are made up of a pair of centrioles and a loose collection of proteins. The centrosome is where the microtubules come together and work together. At cell division, each daughter cell gets one centrosome, which is then copied by the cell before the next round of mitosis begins. This gives each daughter cell two centrosomes. The two centrosomes join tubulin molecules together to help make a microtubule spindle. Further, the centrosomes are then moved along these tubules to different sides of the cell by motor proteins. These were some of the important points regarding prophase of mitosis. Next, we will discuss the other stages of mitosis in detail. If you like the videos, then please do not forget to subscribe to the channel. Thank you and take care.